Okay, so here we're going to look at brain development. Now, embryologically, all central nervous system structures come from something called the neural tube, which is essentially a tube of cells made of embryological stem cells with a fluid-filled center. Now, this neural tube, as it grows and elongates, turns into three primary vesicles, or sort of bulges of this tube. We have the prosencephalon, mesencephalon, and rhombencephalon, which become the forebrain, midbrain, and hindbrain, respectively. Think of the forebrain as like the very tip of the tube. Midbrain is just being a little bit, deeper along the end of that tube, as well as the hindbrain, and then the rest of the tube itself becomes the spinal cord. So if you look at here under A, this is what the neural tube looks like early on in development. So shown in kind of a dark gold here, we would find that these are actually the embryological stem cells that would become neurons and glia later, and there's a fluid-filled center of this tube as well. Now, as this neural tube begins to grow and differentiate, we get the prosencephalon, the mesencephalon, and the rhombencephalon, which are the three primary vesicles here, or sort of bulges of the neural tube. Now, as this neural tube begins to develop, we get secondary brain vesicles. So our prosencephalon becomes the telencephalon and diencephalon. The mesencephalon stays as the mesencephalon, and then the rhombencephalon becomes the medan and myelencephalon, as well as the cerebellum. Now, the rest of the neural tube just becomes the spinal cord. And now these structures here will begin to differentiate further in development and into adult brain structures, where the telencephalon makes things like your cerebral hemispheres. Your diencephalon stays as the diencephalon, which includes your thalamus, hypothalamus, and epithalamus, as well as your retina of your eye. The mesencephalon is the midbrain, and then our medencephalon becomes the pons and cerebellum, where our myelencephalon becomes the medulla oblongata. Now, what's interesting then is that these brain regions are also named for the fluid-filled spaces that they surround. So in an adult brain, we'd find that the telencephalon here, which makes your cerebral hemispheres, surrounds these fluid chambers called lateral ventricles. Our diencephalon surrounds the third ventricle. Our midbrain surrounds the cerebral aqueduct. Our pons and cerebellum, as well as our medulla oblongata, are near the fourth ventricle. And we find a fluid-filled space in the spinal cord called the central canal. So looking at the neural tube in more of a three-dimensional type of view, we would see then that the tube begins to kind of fold and differentiate and start to form these vesicles that are these bulges of the tube itself. So you find the telencephalon, diencephalon, mesen, medan, and myelencephalon. Now, those actually form what we call the brainstem. Diencephalon you find deeper within the brain, and our telencephalon is the majority of the volume of our brain because it forms our cerebral hemispheres. Now, if you start to look here, as the neural tube differentiates and grows, we see the telencephalon starts to get so large that it surrounds and encases the diencephalon, whereas our brainstem stays separate. Now, adult brains have four major regions. We have the hemispheres, cerebral hemispheres, diencephalon, our brainstem, and our cerebellum. So if you look at an adult brain here, you can see that it's even further differentiated, where the telencephalon forms the cerebral hemispheres here that become so overgrown that the brain matter has to form these infoldings, which is why it looks sort of pruned here on the surface. It actually completely surrounds the diencephalon, which is now deep within the telencephalon itself. And then our brainstem here is made of our midbrain, pons, and medulla oblongata. And back here, the hindbrain is the cerebellum. Now, inferior to that, we have the rest of the neural tube, which is the spinal cord at this point. So if you took a cross-section of the central nervous system, you'd find that there's gray matter and white matter within it. Gray matter is darker regions of the tissue, and white matter are lighter. And the reason why white matter is lighter than gray matter is that it's primarily made of myelinated axons that are mostly made of lipids, and lipids are kind of whitish, which gives white matter its lighter appearance. Gray matter is mostly made of non-myelinated neurons and cell bodies, but because these cell bodies contain a lot of organelles and protein, it gives them more of a grayish appearance to them. Now, if you look at a cross-section of the spinal cord here, we'd find that you see gray matter deeper, and it surrounds the central canal, which is the vestige of your neural tube, as well as some white matter around that. Now, if you took a cross-section of the brainstem, we would see lots of gray matter here. In fact, gray matter that's located deep within the brain we actually call nuclei. So these would all be examples of collections of gray matter called nuclei. And if you look at a cross-section of the telencephalon or cerebral cortex, we'd find that out here we have the cortex, which is gray matter on the outer surface of the brain here. And then we also find gray matter deeper within the brain, forming more nuclei, like within the diencephalon. And all of this in between is white matter that's involved with communication of information. Now the ventricles are large fluid-filled chambers that are continuous with one another. So if you go back here and you look at a cross-section of the brain, you see that it's not completely solid. There actually are spaces within the brain, and these would normally be fluid-filled with cerebrospinal fluid. So what happens here is that in the cerebral hemispheres, we have our lateral ventricles, which are located deep within each hemisphere, and they're kind of these large C-shaped structures. Now, each ventricle is also connected to the third ventricle. So the lateral ventricles are interconnected by the third ventricle through this interventricular foramen of Monroe. And then the third ventricle is connected to the fourth by something called the cerebral aqueduct. So if you looked at a picture here of our ventricular system, we'd see our large lateral ventricles found within each cerebral hemisphere. Those are connected to the third ventricle by the interventricular foramen of Monroe, and then your third ventricle is connected to the fourth by the cerebral aqueduct here. Now if you look coming off the fourth ventricle here, we see our central canal, which can continue down the spinal cord, and there's even actually openings on the side, which are called the apertures, that allow for cerebral spinal fluid to exit and surround the outer surface of the brain to provide some additional protective cushioning. So if you look at a side view here of your ventricular system, again, you'd see the lateral ventricles, third ventricle, the fourth ventricle, and then what connects the third to the fourth ventricle is the cerebral aqueduct. Coming off the fourth ventricle, we have our central canal, as well as our aperture that allows for CSF to drain outside of the brain. And then what connects the lateral ventricle to the third is the interventricular foramen of Monroe. Now, what keeps these lateral ventricles separated and they don't communicate with the fluid with each other directly is something called septum pellucidum. And septum pellucidum is a very thin sheet of tissue that separates the two lateral ventricles within each hemisphere.